Hi, welcome to ePathshala. Today's topic is on the concept of glycemic index and glycemic load. The concept of glycemic index was done to provide the numerical classification for carbohydrate foods based on the assumption that the developed data would be helpful in the situations in which the glucose tolerance is impaired. According to Burkitt and Trowell, who developed the dietary fiber hypothesis, suggested that foods that are more slowly absorbed have a greater metabolic benefits in the reduction of various non-communicable diseases like diabetes and coronary heart diseases. Hence, this glycemic index concept was extension activity of the above hypothesis. This module is a major part of micronutrients paper, and this deals with the definition of glycemic index and glycemic load, determination of glycemic index, protocol, classification of glycemic index and glycemic load, factors affecting the glycemic index and glycemic load, concepts and limitations of glycemic index and glycemic load. Thus, this module will help the learner to understand better about the concept of glycemic index and glycemic load. Objectives of this module is to know the definitions of glycemic index and load, its assessment protocol and classification of glycemic index and glycemic load, to understand about the classification of glycemic index and load, factors affecting the glycemic index and load, and limitations of the same. Yes, we must know what is glycemic index. The glycemic index is ability of the test food to raise the blood glucose levels with the fixed amount of available carbohydrate compared to the reference food with the same amount of available carbohydrate. Technically defined as the incremental area under the blood glucose response curve of a specific portion of a test food expressed as a percentage of the response to the same amount of carbohydrate from a standard food taken by the same subject. Yes, now we learn what is glycemic index. Then what is glycemic load? Glycemic load is a function of carbohydrate intake and glycemic index. To say rightly, it is based on the glycemic index and is calculated by multiplying the grams of available carbohydrate in the given food items, the foods a glycemic index, and then dividing by the value by 100. We'll learn the glycemic index and glycemic load. Now we'll discuss about how to determine the glycemic index of a food. The glycemic index of a food is defined as an incremental area under the 2-hour blood glucose response curve following a 12-hour fast and ingestion of a food with a certain quantity of available carbohydrate, usually 50 grams. The area under curve of the test food is divided by the area under curve of the standard and multiplied by 100. A standard either may be a glucose or a white bread. The average glycemic value is calculated from the data collected with a minimum of 10 human subjects. Both the standard and the test food must contain an equal amount of available carbohydrate. The result gives a relatively ranking for each test food. The current validated methods use glucose as a reference food, giving its glycemic index value of 100 by definition. This has the advantages of being universal and producing a maximum GI values of approximately 100. White bread can also be used as a reference food, giving a different set of GI values. For a people whose staple carbohydrate source is white bread, this has the advantage of conveying directly whether replacement of the dietary staple with the dietary food, different food, would result in faster or slower glucose response. A disadvantage with this system is that the reference food is not well defined because there is no universal standard for the carbohydrate content of the white bread. Now a little light on the classification of glycemic index and glycemic load. These values can be interpreted as a percentage on an absolute scale and are commonly interpreted as follows. The foods which have the value of less than 55 are categorized as low glycemic index foods, and the foods with the value of 56 and 69 were categorized under moderate glycemic index, and the foods which have the value of more than 70 will be classified as high glycemic index foods. Regarding the glycemic load classification, the foods with the less than value of 10 were categorized as low glycemic load foods, and the foods with a value of 11 to 19 will be classified as moderate glycemic load foods, and the foods with the value of more than 20 will be classified as high glycemic load foods. Yes, we understood about the glycemic index and glycemic load with the definition and classifications. Let us discuss about what are the factors which affects the glycemic index and glycemic load of foods. In the first factor is the carbohydrate content of foods. Not all carbohydrate rich food result in hyperglycemia when consumed. This indicates that there could be differences in individual components of carbohydrates that are responsible for the repeated variations to postprandial blood glucose response after consumption of the various carbohydrate containing foods by both healthy and diabetic individuals. These variations in glycemic response to carbohydrate foods, and which also tends to affect the glycemic load of foods, were reported to arise from different components of carbohydrates present in foods and their property such as starch composition, dietary fiber, sugars, insulin response, protein contents, processing, variety, particle size, fat and acidity. The next second factor is the starch composition. Starch contributes about 70 to 80% of the total carbohydrates in normal diets. For nutritional purposes, starches are classified on the basis of their rate and extent of digestion into categories, namely a rapidly digestible starch, slowly digestible starch and resistant starch. What is rapidly digestible starch? It is a starch fraction that is rapidly digested and absorbed in the duodenum and proximal regions of the small intestine, leading to a rapid elevation of blood glucose and usually a subsequent episode of hypoglycemia. Then what is about slowly digestible starch? It is a starch fraction that is digested slowly but completely in the small intestine to provide sustained glucose release with a low initial glycemia and subsequently a slow and a prolonged release of glucose, leading to prolonged energy availability. Then what is resistant starch? Resistant starch is not digested in upper gastrointestinal tract but is fermented by the gut microflora, producing the short chain fatty acids that provide additional energy to the body. The third factor is the dietary factor. The American Association of Cereal Chemists defined dietary fiber as a carbohydrate polymers with more than a 3-degree polymerization which are neither digested nor absorbed in the small intestine. According to the American Association of Cereal Chemists, classification of dietary fiber is done into 3 constituents: non-starch polysaccharides and oligosaccharides and analogous carbohydrates, and lignin substances associated with non-starch polysaccharides and lignin complexes such as waxes, phytate, cutin, saponin, tannins and subrins. Viscous soluble fiber play an important role in controlling postprandial glycemic and insulin responses because of its effect on gastric emptying and macronutrient absorption from the gut. Surprisingly, some prospective studies found that insoluble or not soluble fiber was inversely related to the incidence of type 2 diabetes. In clinical studies using fiber supplements, it appears that only the viscous variety of soluble fiber plays a significant role in reducing the postprandial glycemia. This is in contrast to the findings from different authors where insoluble fiber but not soluble fiber from natural food sources was inversely related to the risk of diabetes. The fourth factor is sugars. Glycemic index is affected by the composition of sugar in a food. For example, Sucrose, which is made up of glucose and fructose, has a lower glycemic index than glucose because half of the sucrose molecule is made up of fructose and a type of a sugar that elicits low blood sugar response. In addition, while the glycemic index of sucrose is 68 and the glycemic index of glucose is 100. This variation in glycemic index as a result of composition of sugar could also affect the glycemic load. The fifth factor is the insulin response. This factor that has been found to have a considerable effect on blood glucose level of an individual following consumption of a carbohydrate-rich diet is insulin response. Insulin is a primary hormone the body uses to maintain the blood glucose levels within a healthy range. Therefore, in a well-fed state or when foods, especially with considerable amount of carbohydrates, are eaten, elevated levels of intracellular glucose in the hepatocyte allows glucokinase to phosphorylate the excess glucose to glucose 6-phosphate. This reaction stimulates insulin response or activation, which makes for conversion of the excess glucose to glycogen. The conversion of glucose 6-phosphate to glycogen is favored by inactivation of glycogen phosphorylase and activation of glycogen synthase. Elevated insulin level results in overall increased glycogen synthesis or glycogenesis, and inadequate secretion of insulin could result in improper metabolism of carbohydrates, proteins and fats, leading to hyperglycemia. The sixth factor which affects the glycemic index and glycemic load concept is about the protein content. Protein-rich foods increase insulin secretion, leading to lowering of postprandial glucose concentrations. Thus, the natural protein content of some foods might be the reason why their starches are not easily hydrolyzed, which confers them with the low glycemic index foods. A typical example in the case of pasta in cereals and gluten that slow down the action of pancreatic amylase, thereby leading to lower the glycemic index. The seventh major factor which affects the glycemic index and load is the processing techniques. It has been established that different food processing techniques affect the digestibility of starch, which has some implications on the glycemic index of these foods. Processing techniques may affect both the gelatinization and retrogradation process, influencing the resistant starch formation. For example, roasted and fried foods were reported to have the higher glycemic index than the boiled foods. In another study, steam cooking was reported to aid the production of Rs17, and starches isolated from several steam heated legumes were reported to be rich in indigestible resistant starch, which was not absorbed in raw beans. These reports point to the fact that methods of processing of food samples usually affects their glycemic index. Finally, processing of foods using high temperatures could induce gelatinization, thereby permanently disturbing the amylose-amylopectin structure of the starch complex, making it more readily accessible by the digestive enzymes. The eighth factor is about the variety. Variety is one key factor that affects the glycemic index of foods. For example, the glycemic index of the white potatoes tends to range from moderate to very high, even with the same variety. The next factor is about the particle size. When the starchy foods are ground, their particles become much finer, which make for ease of their hydrolysis by digestive enzymes, thereby increasing their glycemic index. Digestibility of starch is affected by the size of the granule and surface area to starch ratio for action of hydrolytic enzymes. The 10th factor is the role of fat. Fat increases the time it takes for food to leave the stomach and enter the intestine. By slowing the rate at which dietary carbohydrates are digested in the intestine, fat-containing foods may affect the rise in blood sugar and yield a lower glycemic index than similar foods without fat. For example, the glycemic index of potato chips is 57, French fry is 75 and the baked potato is 85. Yes, the next factor which affects the glycemic index and load is nothing but the acidity. Acid in the food slows down stomach emptying, thereby slowing the rate at which dietary carbohydrates are digested. Thus, increasing the acidity in a meal can lower its glycemic index and blood glucose. These factors, as enumerated above, therefore tend to affect the validity and reproducibility of glycemic index and glycemic load of foods. Yes, let us hope you have listened about the factors affecting the glycemic index and glycemic load concepts. Next, we will discuss about the limitation of the concept of glycemic index and glycemic load. The immediate effects of carbohydrates on an individual blood glucose concentration are of interest not only for nutritional guidance, but the glucose concentration has various health implications as well. A major reason for these controversies in the fact that the rate of digestion of carbohydrates varies with the health status, race and gender, and as such the human subject used to test varieties of food for their glycemic index and glycemic load in vivo assays must be carefully chosen. Another argument that has been put up is that the effect of high glycemic index diet is affected by the degree of insulin resistance. Some studies even found greater insulin sensitivity with a high glycemic index diets. Other studies of dietary glycemic index and glycemic load in relation to insulin resistance and the risk of type 2 diabetes also showed inconsistent results. For example, the insulin resistant atherosclerosis study that evaluated the effect of higher versus a lower glycemic index or glycemic load diet on insulin sensitivity in a large epidemiological setting showed no correlation between either the glycemic index or glycemic load and insulin sensitivity. Another problem that has limited the effective use of glycemic index values in therapeutic guide for diabetic patient is that the methods for determining the glycemic index are not standardized, with the values having larger inter and intra-individual variability. For instance, the glycemic index values reported by different authors for potato and rice varied widely. The accuracy of in vivo GI measurement is also influenced by other factors such as method of calculating, IAUC, methods used in blood glucose measurement, defining the amount of tested food which contains 50 gram of hyperglycemia carbohydrates, variability within the same individual and amongst the subjects included in GI determination, day-to-day glycemic variability and time of the day when the test is carried out. And another reason for very different glycemic index values of similar foods are reported by different investigators could be due to differences in starch structure or digestibility, variation in methodology, or to the effects of random variation. Hence, it is suggested that one should be careful in interpreting the results of determining of glycemic index of foods, especially when such results are used as a sole basis for therapeutic recommendations in type 2 diabetes management. Such studies could be confirmed even using the HbA1c analysis assays. We will discuss something about the newer aspects of glycemic index research. There is a considerable interest in the relations between insulin resistance, the generation of reactive oxygen species, tissue damage, and the liberation of pro-inflammatory cytokines and the acute phase proteins, the latter appearing to be a powerful markers of chronic diseases, notably chronic heart diseases. The dietary glycemic index play a major role in this sequence of events. Studies have shown that the postprandial rise in glucose is consistent with the depression of serum antioxidants including lycopene and vitamin E. Presumably, the higher the glycemia, the greater the postprandial depression of serum antioxidants. Finally, supplementing subjects diet with the antioxidant with vitamin E has shown to improve glycemic control. Studies such as these suggest a possible beneficial role for lower glycemic index diets by reducing the oxidative damage. It has been suggested that obesity is related to glycemic index or glycemic load. Studies in altering the glycemic index and load have indicated that the lower the glycemic index and load of the first meal, the less food is consumed in the subsequent meal. Long-term studies are required to define the relevance of these interesting findings. Finally, more studies are required to assess the relation of glycemic index to chronic diseases including cancer, CHD and diabetes. In addition, large-scale intervention studies are underway and more are required to define the therapeutic utility of the glycemic index concept. To summarize this module on the concept of glycemic index and glycemic load, the dietary glycemic index concepts suggest a possible role for the rate of carbohydrate digestion in the prevention and treatment of chronic disease, including those diseases that have been highlighted in the dietary fiber hypothesis and are now associated with insulin resistance. This concept is no longer novel pharmacologic approaches to slowing carbohydrate absorption. Notably, the use of glycoside hydrolase inhibitors are now accepted in the management of diabetes. This above module will help you to learn more about the high glycemic index foods and low glycemic index foods and choosing the best food in controlling the diabetes. So this glycemic index concept will definitely help us to prevent our India from declaring as a future diabetic capital of world.